Good evening, everyone. Can you hear me at the back? Okay. Well, it's great pleasure to be here and talk to you because I love finches very much, and luckily you're a fairly captive audience with respect to your passion for finches. So I'm going to just talk about some of the things that we've been doing over the last nearly 10 years now with long-tailed finches. And very recently, and thanks to some of the people in the room here, we've just expanded this work to include black-throated finches as well. So I'm really just going to give a bit of an overview. So I'm an evolutionary biologist, so I work on a number of different species, but the question that I'm most interested in and that I teach students about at university is about evolution and the process through which species arise. So we all kind of know what a species is, because whatever you're interested in, you can go to the bird book or the field guide, and you can open it up, and it will enable you to identify a species as being something that looks in a particular way and lives in a particular area. And what I'm interested in, and have been interested in for the last 20 years, is the process that generates those species, which is the process of evolution, and it's been taking place over many hundreds of millions of years. So this is just an illustration of the grass finches, and you can kind of see even from this picture here that we've got a number of closely related forms, and so what I'm particularly interested in are how we're moving over evolutionary time, and in this case over within the last 10 million years, we've got this divergence between these different forms of things. So the kind of questions that we ask in my research lab are, firstly, what is a species? And I'll come on to why and what I mean by that in a minute. Then, most importantly, how does a species evolve? And how do species interact with one another? And how might that affect processes like their behaviour and also their reproduction? And that might be something that you're particularly interested in. And then finally, there is some value in, although I'm just fascinated by this work and I'm very lucky enough to do something that I'm very passionate about, there is value in understanding these questions will help us to manage and protect species, and will be particularly important in things like captive breeding programs. So as you probably know, so this is a picture of Charles Darwin, a real hero of mine. And Charles Darwin, in his book Origin of Species, was the first person who really laid out a way of understanding how species evolve. And this is a very famous diagram where he actually sketched in his notebook, I think, and then he drew this diagram. And what this diagram is showing us, this was his kind of eureka moment where he actually started sketching out the idea that all these forms that are out there that look kind of similar to one another are related, and they're essentially joined as on a family tree. So this was a very influential sketch of his, and then of course he worked over about 20 years putting all of his work together into a really amazing book which has stood the test of time, even though at that stage of course we knew nothing about genetics. Now in terms of what a species is, so again these are just some pictures from one of the bird guides. And again, species aren't necessarily that clear. So particularly in Australia, when we go out into the field, we can find lots of different races and lots of different subspecies. And again, all of you lot are very familiar with this, with the birds that you're keeping and breeding. So again, what this is telling us is that species aren't necessarily that straightforward, and as we move across the landscape, a lot of species are changing slightly, and there's potentially gene flow and interactions between these different races and different forms. So these are just a random collection of different bird families in which we have a lot of morphological and colour variation across the range. And again, we're interested in what processes formed that variation in the first place. In some cases, this variation was formed tens of millions of years ago, but also what preserves this variation. So why do we still have these different forms out there? Now coming back to Darwin, Darwin should also be a hero of yours because he was also a really super passionate aviculturist. So Darwin kept many hundreds of pigeons, and it was his work on pigeons, and these are just some of the fancy pigeons that were a really big thing in Europe and in Great Britain at the time. He was thinking about his how species change over time. The work he did on pigeons really influenced his understanding of how species more generally evolve, because he realised that if man can do this to the rock pigeon in just a few hundred years, or in fact it was even less than that, he realised there must be some stuff going on. Of course, he didn't understand the genetics, but a similar thing can change one species into another over a period of greater time. So this is the main book, The Origin of Species, and chapter 1 in that book is all about variation under domestication. So again, there were actually 2 or 3 chapters in this book that were just describing all this amazing variation. And of course, aviculture, not just for pigeons, but more generally in birds, was pretty big in the mid-1800s when he was working. Now, essentially, where we're at today, and again to introduce now what I've been working on, is so we've moved forward from that tree that Darwin sketched out, and we can now basically infer for any group of animals, we can infer the relatedness between them. And now, something that Darwin couldn't do, we can actually back this up with some genetic or molecular data. So what we can do is we can actually date the point at which any 2 forms of species or subspecies in this case, we can date the point at which they shared a common ancestor. And so these dates here, this is 0.6 million years ago. That was the point at which the black-throat shared a common ancestor with the long-tailed finch. So 0.6 million years ago, there would have been something, we don't know what it looked like, but it would have been the common ancestor of all 3 of these forms. And then 0.3 million years ago, that's when there was a common ancestor between the yellow and the red. And again, we don't know whether that was a yellow bill or a red bill, but we do know that they diverged around about 0.3 million years ago. So these are actually incredibly recent species in terms of avian species. And this work has all been, these dates have been estimated on the basis of a lot of molecular data. So what we think has happened, and again, we haven't proved all of this, but the reason that we believe this is true is because there's lots of species that are split in roughly the same way as the longtail and the blackthroat across the top end of Australia. So essentially about 1.5 million years ago, there was a continuous band of suitable habitat. So the habitat needs of these finches, the grass finches, would have been very similar today. So open savanna woodland would have been in a continuous belt across the top end of Australia, much like it is today. But we know from a lot of work in paleobotany and geology, we know that Australia dried out. So by about 500,000 years ago, it was at the extreme of its aridity. And what that did, it pushed that savanna country right up into the 3 tips of northern Australia. So there was a very small patch up on the Kimberley Plateau, which was just a very small, relatively small patch of suitable country for all of the grass finches that now live in the savanna. And lots of reptiles, things like red-backed wrens, lots of other species that are dependent on this habitat also got pushed into that refugia. Then there's the Arnhem Land Plateau and Cape York Peninsula. So essentially, probably for around about 500,000 years, we had populations of all of these different species that were completely isolated from each other. And then over the last 300,000 years or so, the country has, or the desert has receded again into the centre of Australia, and once again, all of those remnant or refugial populations of all of the animals that live in this habitat have joined up once again. Now, for some of these species, as they've joined up, there's been enough movement across those, across the whole top end of Australia, that we don't see any evidence of this process. It's just been completely wiped. But for some of the species, and the longtail and the blackthroat are a really nice example, but there's lots of others in all animal, different groups of animals, we have the three forms today that reflect the three refugial forms or populations that would have been stuck in those ends. So essentially what we've got is that as they've come back into contact, which would have been in the last few 100,000 years, as they've come back into contact, they haven't completely broken down the barrier that formed between them. So if we go somewhere up in the area just to the, it's actually just to the area just to the west of Catherine, we find a really distinct join between the yellows and the reds. And so again, the question that we're asking is, why does that join, or why does that barrier still exist? Why hasn't this speciation process broken down? So what are the processes that stop these different morphs or subspecies, if we include, or species rather, if we include the black-throated finch? What processes stop them breeding with each other over the last few 100,000 years? What are the specific genes that are involved in the differences between these subspecies? So we know there's obviously some genes, for example, the genes that are driving bill colour, because you know that if you try and breed these things, they breed relatively true to form. So it's these questions that we can get at in these finches particularly that help us understand what drives the formation of species. And this is still a really big question all over the world. But we're in a really good place to answer this with these species. So as I say, I'm incredibly lucky. So we've done a lot of work out in the field right across the top end. This was a group that I had working up in Wyndham, where we did a 3-year very intensive study of long-tailed finches. But as I'll show you in a minute, we've also worked right across the top end doing a lot of sampling. And as well as doing all the work in the wild and looking at that variation, we've also been extremely lucky that we've got a nice facility at Macquarie University where we can also breed a relatively, or a reasonably large number of these birds in captivity. And so that enables us to go out into the wild and measure in great detail what's going on. And then we can bring them into captivity, we can do experiments, and we can understand what the underlying mechanisms are. So I'm just going to run you through a little bit of that work. Before I do that, I'm just going to try and run through some of the underlying genetics. As I say, Darwin had no understanding of this, but over the past 70 years or so, our understanding of genetics is actually really incredible. Where we're at today in terms of understanding how genes work and how genes are distributed across the genome. So essentially what we're looking at here is in those periods of time, which would have lasted several, probably 100,000 years or so, when you've got those three different populations, over that period of time, two processes have happened. One of them's completely random. So every time that animals breed, there are mutations that are fixed in subsequent generations. Most of those mutations you don't see, but of course you're familiar with some of those mutations, because some of those point mutations will lead to differences in appearance. But because of the size of the genome, which I'll show you in a minute, there may be mutations that are occurring in those populations, and by chance, because you've got three different populations, they will accumulate different mutations over thousands of generations. So that's a completely random process, and that's called genetic drift. The other thing that will happen is in those three populations, the local environment is different. So we know that in Australia, it's actually wetter on the east side of the top end than it is on the west side. So that means that there'll also be selection so that some genes will be differently selected in those 3 different populations. So together this means that the longer populations are isolated, the less likely it is that when they come back together, they will be compatible. Now the genome, we now understand, is absolutely huge. And what the genome is, so the genome is all of the whole collection of DNA that an organism has, and as you know, the DNA carries the instructions for making the proteins that build an organism. So just as an example of how much information is kept in the genome, the human genome, which has been thoroughly sequenced in numerous human populations, has 3.1 billion bases. And so those bases, or the combination of those 4 different bases along the strands of the DNA, are coding for everything, and there's also a lot of junk in there as well. But the thing that's really incredible is that every single one of your cells contains all of that information. And what's more incredible is that all of that information, 3.1 billion letters of information, is all copied every time a new cell is produced. So that's the kind of information that's being copied, and that information, as you probably all are very aware, is packaged across the genome. It's packaged in chromosomes, and this is a carrier type of the avian chromosomes, and the chromosomes are of different sizes, and that becomes important later on. And the other thing to note is that in birds we have the ZW, and again you're all familiar with that because you use that as part of that toolkit for sexing your birds. There are 2 copies of each one of these chromosomes. One comes from a mother and one comes from the father. And again, that's important in terms of the way that things can evolve, because during the production of sperm or eggs, there is recombination of the material on each of those chromosomes. So each male in a population has one chromosome that comes from its father and one that comes from its mother. But as it produces sperm itself, or eggs for the female, each one of those chromosomes will actually swap a section of the chromosome with the one from the mother. So that means that all of the sperm that are being produced by a male are actually slightly different from, they have a different set of DNA in them to the DNA that the male himself has. So this process of sexual recombination, or recombination of chromosomes, is why animals actually produce sexually rather than asexually, because it means that the offspring aren't exactly like the parents. And the reason that this is important is because it generates a lot more genetic variation than clonally producing animals. So getting back to the long-tailed finch, what we've been looking at over the past 9 years or so, as I say, is in the long-tailed finch to start with, but more recently in the black-throated finch, we've been taking these 2 subspecies and trying to understand that genetic variation and some of the consequences of it, both in the lab and in the field. So this is a picture of 2 birds in our aviaries, and these birds were taken from the wild in 2009. So the total sampling that we've done goes from around about the east. So the dotted line here is the range of the species. So we've sampled right from the easternmost part of the range, almost to the most western part of the range. And these are all the populations that we've sampled, and in each population we've sampled around about 100 birds. And so for that sampling, all we've done, we've just basically measured the birds in the field, we've banded them, taken a blood sample and released them. The birds that we've been breeding in captivity, they come from population number 1 and population 17. So those birds come from essentially a red population and a yellow population. And in terms of the variation that we see in the wild, there are two different lines here, and they represent the variation that we see in bill colour, which is this red line here. So as you go across the whole part of the essentially the easternmost half of the range, there's no variation at all in red bill colour. So they're just as red in Catherine, or just to the east of Catherine, as they are over in Mount Isa. Okay, so there's no real variation in the redness of the bill. Similarly, if you go right across to near Derby and you come right across to Tennant Creek, there's no variation in how yellow those birds are. There's just a very narrow band geographically, which is 125 kilometres long, where the population changes from yellow birds to red birds, and in that area, the birds are orange. Okay, so it's a very clean break. Because that area is so narrow, that suggests that there's not gene flow from one side to the other. Otherwise, we'd expect all of these birds to be orange, probably all the way across, or we could have a continuous cline. So just the narrowness of this band actually suggests there's a lot of selection that's preventing the hybrids from successfully breeding in the wild. So the blue line on this same graph, so the distance here is measured from west to east. The blue line also shows that there's a very steep cline, and that cline there is in the genetics, and particularly the genes on the sex chromosome. And I'll come to that. I'll come to that in a minute. So again, the scale of the genetic data we've been looking at in these species, and we've also looked at the zebrafinch and the double-barred finch as an outlier to construct these trees. So we've actually genotyped 44 million SNPs in the zebrafinch and the double-bar, and 26 million SNPs in these two species. So each one of those SNPs is one of those base pairs. It's a single point on the genome in which there's there's two different bases. So in some populations there's a C, which is one of the bases, and in the other one there's a G, which is another one, which is an alternate one. So we've got a huge amount of information, and that enables us to build these trees and to date the divergence between the species. More importantly, that now enables us to look across the whole genome and all of those different chromosomes and look at where that variation is occurring. And that can tell us something about the genes which are being selected and keeping these two forms apart. So these three different figures are essentially just comparing the difference. So if we move up and down, that's the difference between two populations of all these genetic markers. So on the top, we're comparing two populations of the yellow-billed longtails that are 246 kilometres apart. So essentially we're looking at what genes differ and where those genes are across two yellow-billed populations. And then the same here for two red-billed populations. And then down the bottom, we're looking at the difference across that very narrow 121 kilometre hybrid zone. Across the bottom, we're looking across the whole genome, and particularly we're looking at the different chromosomes on which all that genetic information is stored. So what we can see, so anywhere where we have a peak, that's one gene which is really different between these two forms. So as you can see, across the whole genome, so across all these different chromosomes, there are a few peaks or genes which are significantly different between these forms. But the most striking thing here is the Z chromosome. So the Z chromosome is here. So these are all the other chromosomes. The colours here represent the different chromosomes. The Z chromosome is where nearly all the action is. And this is the sex chromosome, that males have two copies of the Z and females have a Z and a W. There's nothing very interesting happening on the W chromosome, but look at all this difference on the Z chromosome. So it appears that most of the difference between species is primarily carried on the sex chromosome, with just a few other points across the whole chromosome, across the whole genome, where there's something kind of interesting happening. And again, we can map those differences according to the different chromosomes. And again, you can see here there's a very steep line. So there's lots of interesting stuff happening in the area just to the east of just to the west of Catherine. That's where you get that really big step in Z chromosome variation. And then we've actually included the black-throated finch on here, and we have another breakpoint as we move from the Z chromosome of the long-tailed finch to the Z chromosome of the black-throated finch. So again, the same story. All the interesting stuff is happening on that sex chromosome. And again here, this is the same diagram at the top, but now we've expanded the Z chromosome, so we're looking all the way across the Z chromosome, and what you can see here is just how much of the Z chromosome is different between these two species. And so we believe this is probably something, so this has been found in a couple of other bird species, so we believe there's something really special about Z chromosomes, which mean they're super important in the formation of species. So the obvious question there is what genes are on the Z chromosome, which might be driving its important role in speciation. And the obvious candidate there is that genes to do with reproduction and maleness particularly are the genes that are likely to be mostly on the Z chromosome. Now, although in the long-tailed finch both the males and females are similarly coloured, an obvious thing to look at, because we know that they're different from the bird books and from all the work you guys have been doing over the years, we know that there are genes to do with bill colour, and so we can just run these kind of experiments. And these are things that some of you have probably done decades ago in your aviaries. So we can ask questions about what happens if we mix, or if we pair a red bill and a yellow bill. Will we get a set of orange birds? Will we get a split between some reds and some yellows? And then what happens when we breed the resulting hybrids? What will we get in the next generation? So we've been doing that over the last few years in the aviaries, and on this graph we've essentially plotted colour in three-dimensional space, and what you can see here is that the yellow bills are obviously yellow, so this is where yellow is in three-dimensional colour space. The red bills are red, and that's not a surprise. And what we find is that the hybrids, which are the orange birds, they're roughly intermediate, but then the back-crossed, so the F1 hybrids back-crossed with the parental birds, they show much more variation in their colour. So even within a family, some of those back-crossed young are completely yellow, some are completely red, and some are intermediate. And that suggested to us that there's 2 or 3 genes at least involved here, and that those genes aren't all, and we know those genes can't all be Z-linked because of the female expression of colour. So again, we've gone back to those plots that are looking across the genome, and we've zoomed in on the differences between the 2 different forms. And what we can then do is we can identify, or what we have done is we've compared the birds of different bill colour, and we've identified 3 different genes which are significantly different across the reds and the yellows. So these names are technical names. These genes have all been found previously in other animals, and some of these genes are extremely old. So some of these genes have been found in basically things like worms. So some of these genes have been around for hundreds of millions of years, but in this particular system, they're driving the variation in bill colour. And one of those genes, that CYP2 gene, had been shown about a year ago to be involved in the yellow bill mutation in the zebra finch. and that's a mutation that's very commonly found in captive populations, but it also occurs in the wild in the Timor subspecies of the zebra finch. So we're now at a point where we can understand exactly what the colour, the genes, the genetic basis of colour in this speciation process. Now, the other really obvious candidate for selection across one of these species boundaries is stuff to do with sexual reproduction. And again, the most male trait that males have is their sperm. And there's a lot of selection on sperm because a male will produce millions of sperm and only one or two will get to fertilise eggs. So again, what we've been doing over the last 3 or 4 years is measuring sperm in great detail and essentially measuring the morphology in particular. And this is a picture of some avian, these are pictures of long-tailed sperm, and we can measure the size of the head. So that's the head that contains the genetic material that's going to be passed on. The mid-piece contains the mitochondria, so that's essentially the engine for the sperm, and then the flagella, and that's just the tail which beats and helps those sperm to move up to the point of fertilisation. So we've measured those sperm across those 2 subspecies because we know that sperm vary a lot across different species of birds, so it seems to be something that could help to keep those 2 subspecies separate. And indeed what we've found is that although it's very subtle, there are significant differences in the size. So red-billed long-tailed finches have larger mitochondrial mid-pieces than yellow bills. So there's a significant size difference in sperm. Then the next question was to what extent that might affect fertilisation when we do experiments by interbreeding these birds across this in the lab. Essentially, if we create these hybrids and then do a back cross between the hybrid and one of the parentals. This is the back cross, which in the wild is what's preventing that zone of contact becoming wider. It's because the hybrids that are being produced aren't successfully reproducing. So that's what we've been able to do in the lab. And one of the nice things about birds is that we can actually go in and count the number of sperm that get onto the egg membrane, and that gives us a measure of how effective a male is in a particular pairing at getting sperm to the point where it actually counts. Okay, so this is a freshly laid egg. We can see the germinal disc, and around that germinal disc there will be a few hundred sperm, which are essentially the also-rans, the sperm that nearly fertilised that egg. And the number of sperm that are found on the egg predicts how many of those eggs are likely to hatch, and that will of course influence reproductive success. So again, what we can do, and we've done this, we've actually used these methods to assess the fertility of birds, which is something that we're going to hopefully do later on with some looking at inbreeding and outbreeding in blackthroat populations. But essentially what we can do is we can dissect the yolk membrane off of the yolk, and here we've got an infertile egg on the left, and we can stain that egg membrane, and here you can see there's just 1 or 2 sperm that are stuck on that membrane. On the right, this is a fertilised egg, and that egg, the germinal disc, has hundreds of sperm stuck in the yolk membrane. So we can essentially assess the quality of a pairing in any context, but in this context across those different subspecies, and we can say how likely are they to have actually resulted in successful reproduction. And what we've found is good evidence that the hybrids, so the orange birds, when we back cross them to one of the parental forms, there's a lower number of sperm getting to the egg membrane. So that means there would be filters. There's probably nothing wrong with those sperm themselves, but there will be filters up the female reproductive tract that prevent sperm from the wrong male fertilizing her eggs. And those, if she is in a position where she's copulating with multiple males in that contact zone, her eggs are far more likely to be fertilized by the parental forms. And incidentally, we're also looking at, we're using these same techniques actually to also assess the quality of pairs. Even in things like zebrafinch, we're finding that pairs that have stayed together for longer and are better breeders also have better numbers of sperm getting onto their egg. And that might reflect behavioural coordination between the partners. So that's kind of what we've been doing in the longtails to date. And then very recently, over the last 6 months, we've brought black-throated finches into the equation. Now, the reason we've done this is twofold. So the first thing is that the divergence between the black-throated finch is about twice as old as it is between the red and the yellow longtail. So that means the effect should be a little bit stronger. So that's one of the reasons that we want to look at the black-throated finch. But the other reason is that the black-throated finch, as you know, is the focus of a lot of conservation work, and there have been suggestions that at some point in the future there might be a need for some captive breeding programs. One of the big questions around this whole compatibility issue is, again, even in the black-throated finch, we know that there's variation genetically across the population from north to south. And one of the questions is, do we have the same kind of incompatibility issues within that species? And also, will we have those incompatibility issues when we start breeding blackthroats in captivity, if the level of inbreeding gets to a certain level? So those are the reasons that we're also including the black-throated finch. So we've characterised the long-tailed finch pretty well across the whole of its range. So in the next year and a half, the plan is we're going to be doing a similar amount of genomic work on the black-throated finch from the samples that were collected historically and potentially by aviculture in the south of the range right through to the north of the range. So we're going to characterise the genetic variation in the blackthroat, exactly the same as we have for the longtail. And then we're also going to look at the interaction between these two species. So we're going to look at the population genetics across the natural range, but also we're very interested in looking at the genetics of captive populations. And I've spoken to some of you about that already, but that's something that we're going to hopefully, with help from you guys, we're going to try and drive that project through in the next year or so. And then we're going to explore these incompatibilities, which will help us understand to what extent some of these incompatibilities that we're showing might be a problem in any avicultural population, but particularly for things that might be the focus of captive breeding programs. Okay, so I'd just like to acknowledge Laurie, who's going to come and talk to you in a few months, who's been working in my lab for 5 years now. Daniel Hooper, who's doing a lot of that genetics at Cornell University in the States. Callum McDiarmid, who's working with me as a student at Macquarie, and then Melissa Rowe, who's working in Norway and who runs a lot of that sperm work. Okay, any questions? It was all on the slides. Yep. Yep. So that's a really good question, and that one of the big challenges we have, and the reason that we didn't include the blackthroat originally is because as soon as you start thinking about how you're going to do it, you end up with lots of different combinations. So to date, most of the work, so we have used both the cock and the hen as the hybrid, the F1 hybrid in the back cross, and then obviously the other one's the other. But the sample sizes today are pretty small, so we haven't been able to separate out whether there's a bigger effect of whether it's the cock or the hen. There doesn't look like there's a difference there. But look, it's a really good question, and so over the next year we're going to improve those sample sizes and try and because there's good reasons to think there would be mechanisms that would operate differently across those different things. And we also, what we haven't done yet, but we've done all the sampling, and we took the last photographs today. So we've also measured sperm from, I think, about 90 hybrid males and 90 back-crossed males. And so we're measuring the sperm of those to see if it's the sperm that are compromised, because they could be malformed or even shorter or something like that. Yeah.
Sorry, if you had a hybrid between the two there, some of those species will reproduce. I'll give you an example, the siskin from South America and the canary. I believe that when they originally did that, it was a canary hen and a siskin male. Now, is there a difference in doing that with a male and a female?
There may be. So there are lots of species where other people have looked, particularly at things like ducks, and a lot of there's not so much data on finches, but there are people who have looked at what the chances of producing viable hybrids are, and a lot of it will depend on how long those things have been isolated. Now, some species, even if they've been isolated for 10 million years, they can still produce viable offspring. And what we predict, so part of the reason is you would get selection that would be stronger when they're back in contact. So if you take two things that have been completely separated, they actually haven't had selection to reinforce that boundary. These things have. So a lot of those differences that keep them separate will actually happen quite fast once they come back into contact, if you've got those genetic incompatibilities. And whether there's asymmetry in terms of whether a female is more likely to breed with a male than the other way around, that hasn't been explored in enough species to know.
Yes, because it's rather strange. Like, you can look at some of the Australian parrots, say, Turkazines and Scarlets, and they'll hybridise, but none of the offspring will be fertile. Now, from what I've heard, the hens never lay. But the cockbirds are infertile in itself, so they're both infertile.
So usually
they look the same.
So there's a very clear effect called Haldane's rule, which is that in the heterogametic sex, which in birds is females, the effects will be twice as bad as in the male, and that's because they only have one. So if you've only got one copy of a Z, so males have got essentially two different copies. And so one of them's more likely to work. So it's definitely true that females typically show greater effects of sterility and things than males do. Some of the things that we're also looking at is the chromosomal structure, and so some of these things, it's not just how long they've been separated, but some other chance things, like you can get whole chunks of those chromosomes which actually flip round during that process of recombination, and that can very quickly lead to a big incompatibility. So again, that's a very chance effect. So it means it's not a perfect relationship with how long they've been separated.
I think there's actually quite a lot of examples we could probably bring up in aviculture where it's the cock from one species with the hen from it's got to be that way. The cock from that species with the hen from that species to get any young at all, let alone viable young. You do it the other way around, it doesn't work. And other species will be different, won't it? And I'm sure there's some of the Maltese friends here would have examples, and maybe Joe's going to give us some now. He's got his hand up. Hang on.
My question is that do the female hex and the longtails are promiscuous? Or they pair bond and they stay true to that male?
Yeah. So that's a really good question, and we are looking into that. So one of the things, so typically they're not that promiscuous as a species. So there's not the level of extra pair paternity in wild populations is around about 15%, which is kind of average. It's not up the high end. And what we would predict is that in that contact zone where a female is at a risk of pairing with the wrong species, because she doesn't necessarily know what her genetic background is. But in that contact zone, females should be selected to actually be more promiscuous. Because what we do know from other species where we've done experiments is that if you copulate with a male from the right species and the wrong species, the sperm from the right species is far more likely to fertilise your eggs. So there's been experiments, we've done experiments in Gordians, there's been experiments in chickens and ducks that have shown that, and fruit flies and mice. So that seems to be a fairly well-established pattern. Yeah.
Are the young'uns of that male? Or are they two young'uns of that male and one young'un of that male and another?
Yeah, so in the wild, that's exactly what happens. So 15% of all the young in the nest are not fathered by the male who's rearing them. And in some nests, we have 3 males. There's 4 chicks in the nest, and 3 males have fathered them. What we're now doing is we're now going back and looking at, so that was data that was collected in Wyndham, which happens to be around that chromosomal contact zone. We're going back and we're looking at those different nests, and we're seeing if that's because the female mismatched with the male partner at some of these important genes. So absolutely it does happen. Yeah.
Any others? I was just going to ask, in terms of the orange-billed birds, is it a continuum from yellow to red, or how does it seem like you've got orange, yellow, or red?
Yeah, so in that first generation, the hybrids are pretty much all orange. And it's only when you start back-crossing them, or even breeding the hybrids with themselves, that you then start getting the others flipping out. And that's because there's multiple genes there. That means some of those offspring there's the potential for some of those offspring to have completely a yellow set of genes.
So it's not sort of a straight dominant recessive sex-linked. It's a combination of those for each of the 3 genes.
It's a blending of those different genes,
which makes it.
Because there's 3 genes, you can get multiple colours. So we don't know exactly, at this point, we don't know exactly. So we know the 3 genes that are involved. So we know there's these 3 different genes involved, and each one of these genes has 2 different forms. So to get a yellow, you would have, presumably, but we haven't actually done that test yet, you would presumably have the yellow copy of all 3 of those, and to be a pure red, you'd have the pure red copy of all 3 of those. So anything else, the oranges might have 2 copies of the red and 1 of the yellow. But the other thing is we don't know if all of these genes are adding the same amount of red or yellow. It could be that one of them is stronger than the other one.
Why I ask is that I'm sort of interested in birds that are true to the wild type.
Yeah.
And my preference would be to avoid orange birds.
Yeah.
So I'm guessing you're saying there's various shades of orange.
Yep.
And if you take that back to Gouldians, where there's less and less normal Gouldians, and it's hard to tell by looking at them, is there some way in the future where you will be able to work out what's a normal Gouldian and what isn't?
Yep. Yeah, you should be able to. So we're very close to doing that now in a lot of birds. As I say, there's increasingly, because there's more and more genomes becoming available. So this is only about the 6th avian genome that was completely sequenced. But even though we did that about 5 years ago, there's now hundreds of avian genomes. So hundreds of species have now been fully sequenced. And we're finding that some of these genes are the same across lots of different birds. And interestingly, it's not all. So this particular gene is involved in changing carotenoids from a yellow to a red state. This particular gene, but this gene is involved in other types of colour. So it might be involved in structural colour, for example, in the Gordian between a blue and a white. And so within the next 10 years, you will absolutely be able to do that. I predict because enough of this information is coming online, and it's now relatively straightforward.
At an affordable rate. So we'll be able to send off a blood sample or whatever and come back and say, yep, that's 100%
yellow. Probably,
blood.
Almost certainly.
So am I right here? Is there really only like 2 to the power of 3, 8 possibilities, given there's those three genes? Is it
so essentially the next thing here, so we've already got all the measures, we just need to... so here are all this is the colour space, and you can see that there's a lot of variation in those back crosses and the orange birds. And essentially what we will do, because we've got the DNA from all these birds, we'll be able to say, okay, this bird here, we can sequence those 3 genes, and we can say this particular orange comes from a combination of one of those CYP genes and one of those TCR genes. And likewise, we can then say this bird here is this kind of orange because it has the other. And we'll also then be able to say which of those 3 genes has the greater effect. But it also, the other thing to just note against that is that a lot of these things aren't necessarily straightforward, because you can also have control genes which aren't the ones that we're sequenced. There's also these other genes here. So these genes are the ones that we've identified as definitely being involved in the colour. But there's all these other genes. Some of these might be controller genes, which are exerting an effect on the expression of these ones. So it's not absolutely black and white. So it's going to be a little bit more complicated. Yeah.
In the future, are we going to have a third subspecies? Is the yellow ones in the band the creation of another subspecies?
Which ones? The yellow ones?
In that 120 kilometre strip?
No. So the reason that probably won't happen is because so this is what's really happening out there. There's gene flow from these other populations that's getting in, and that's breaking down. So within that strip, there's probably nothing that's going to... so you've got a lot of pressure from outside, because there's millions of birds on the east and the west of that strip, and they're constantly pouring genes into that area. And at the moment, although most of those hybrids are essentially dying off, or their gene lines are dying off, because they're breeding and they're not producing any viable offspring. So there shouldn't really be anything that's going to cause those birds to become separate. What will probably happen with climate change over the next million years is that the whole pattern will shift again. So they'll go back into refugia probably, and they'll come back out. And that's why if you look at all the grass finches and a lot of other birds and reptiles, they don't all have 3 different forms. Some of them have got 2 forms, like the mast finch, and presumably the mast finch would have also gone into these 2 refugia here. They would have gone partly down the same path, but for whatever reason, when they've come out, that split there has broken down. So everything's kind of possible, and it kind of depends on what happens, how much change there's been during the time that those have been separate. So, and then there's other things like the Gordian finch. The Gordian would have gone into these same 3 refugia, so it's almost certain that the 3 head colours evolved at exactly the same time as these ones, in the same 3 refugia, but they've come out, and because they're a more mobile species, and because of the genetic architecture of those head colours, you've got a different process. So everything's kind of possible. Yeah.
Sorry, I'm just starting the camera again. Any other questions anybody would like to ask?
So how many people here keep black-throated finches?
Oh.
A few? Okay. So some of you will be aware of... so we did a similar project previously with Gordian Finches, with Perry, who came and spoke to you probably over a year ago now. So we are writing that work up. Hopefully we'll be able to communicate that to you in the next few months. Those results have been finalised, but Perry went to the States, and she was there for about 6 months. She recently came back to Canberra, and I think she's got lost in an admin job in Canberra, just to keep some money coming in. So she is writing it up. But essentially what we found in the Gordian Finches, if you remember, a lot of you, or some of you, would have donated some feather samples to that. And for that, we were really interested in what the genetics... So we've already done the genetics of the Gordian Finch in the wild. We were interested in what's the genetics of the domesticated population. and we found really significant genetic structure both between the east coast and the west coast, which probably won't be a big surprise. There was also significant structure between the redbirds and the blackbirds in the captive population, and there was a little bit of structure down the east coast as well. So I will definitely report that back to you at some point, either through something for the magazine, or someone will come and talk to you about that. What we're interested in doing for the blackthroat is, again, we're interested in looking at using the museum's specimens, and also any specimens that people have that they think represent the old populations, particularly from the south and the north. We're interested in characterising the genetics of the wild populations, but also the captive populations, to see how well those marry onto each other. So again, hopefully in the next 6 months we'll be in touch, and it would be great to get some samples from some of you.
Yeah, if anyone's wants some blackthroats, actually Renee said she's got something like 20 pairs at Ace Colony at the moment. I'm not absolutely sure where she got them from. She didn't get them from you, did she? She didn't get them from me. I'm not sure where she got them from. I've also got quite a few that I'll have to get rid of. Probably got, I don't know, 30 or something, maybe. I've got to cull down again too. So if anyone's interested in getting some blackthroats so they can contribute, there's a few around. If you'd like that. Mine, I've got a Parsons, and I know the ones that Renee's got at the moment are Parsons, which are the southern ones, the white rump ones. But there's certainly people with the black rump ones that we call diggles as well, the northern subspecies. So yeah, by all means, get involved. Any other thoughts or questions? Thank you, everyone, and thank you very much, Simon.
All right, thank you.
Thanks everyone. Brian, I've stolen your microphone, mate. Well, I bring it up.